Hello, can everyone hear me?
Yes, we can.
Uh,
Can hear me? Can you hear me, Ryan?
I can hear you loud and clear.
Wonderful.
I just want to apologize for everyone for the technical issues that we had. I had trouble logging in, and I just got a good start on practicing the presentation. The last 10 minutes, 5 minutes, I've been talking on the backstage. So I apologize for that, and we're ready to go whenever you are.
Awesome. I'll just quickly introduce you here, Ryan, and then let you get to it. So Ryan's presentation is on enhanced biological phosphorus removal. Ryan is the principal scientist for Ryan Hennessy Wastewater Microbiology. Ryan is a certified wastewater operator with 15 years of hands-on experience and over 10 years of consulting experience. Ryan was mentored by Dr. Michael Richard, and when Dr. Richard retired, he inherited an existing client base, allowing works as a full-time consulting microscopic evaluation. Ryan has spoken at many conferences and held many training sessions over the years and takes pride in presenting in a matter that hopefully reaches a broad spectrum of audiences. All right, go ahead, Ryan.
All right, thank you. I'm going to share the screen here and have my presentation up here. So with that said, we'll get started. This slide here is just a general outline of what we're going to be talking about. And I'd just like to start off with, with the phosphorus limits. And I'm not sure, I'd imagine they're coming to just about everywhere in the northern in North America. In Wisconsin, in the last 10 years or so, where I am in the United States, the phosphorus limits have become increasingly stringent, and a lot of plants, depending on where you're discharging to, have limits as low as 0.075 milligrams per liter. So in the in that process of increasingly stringent phosphorus limits, plants have begun to get more creative in terms of what we need to do in terms of removing phosphorus. So with that said, a lot of plants are using tertiary filtration and also enhanced biological phosphorus removal. So just a really quick explanation of what we're after in enhanced biological phosphorus removal is if you take the average microbe that we have in standard activated sludge system, they're going to vary a little bit, but on average, they're going to make up about 2% phosphorus on a dry weight basis, where your PAOs or your polyphosphate accumulating organisms can consist of as high as 35% phosphorus by weight. So the goal for enhanced biological phosphorus removal is to create conditions in the treatment plant where we can harbor a stable population of these bacteria that have a higher affinity, a higher amount of storage capability for phosphorus. And if you have a stable population of PAOs, generally this is going to get your sludge content of phosphorus as high as anywhere between 4 to 7%, 6% here on the slide on a dry weight basis. So there's the BioP bacteria. We have a lot of genetic diversity within PAOs. And in terms of this, I would say Accumulobacter and Tetrasphaera are our predominant PAO genera that we're going to traditionally see. We have a couple others, such as Dichloromonas, which is a GAO and also a PAO, and it has variable capabilities. So what's very interesting is the Accumulobacter is most often correlated with the traditional PAO phenotype, which is under the microscope, we see Neisser positive clusters of cells. And when they're storing phosphorus, they present Neisser positive staining characteristics. So if you have a sample that you're looking at and you have PAO, but they have released phosphorus, you're not going to see the traditional PAO phenotype under the microscope. So it's only when they're storing that polyphosphate that you see it. Now you have your traditional PAOs, and then you also have the non-traditional PAO phenotype, which is incidentally a nostocoidal filament morphotype. So tetraspira is very often in a filamentous form. So when we are looking at under the microscope, assessing relative PAO abundance, it's really important to note that we are only looking at the traditional PAO with the Neisser positive staining characteristics. And tetraspira is very often a filament, a nostocoida limicola filament type. So you can make some general assumptions. You're safe to say there's enhanced biological phosphorus removal occurring if you do see the traditional, but in the absence of the traditional PAOs, you could still also potentially have biological phosphorus removal. Another thing I want to bring up is we have 17 different species of tetraspira. And then when we look at our other PAOs on the Accumulobacter side, we have 12. And on this other one, which is pretty rare, we have 2 different species. So the biogenetic diversity of PAOs is a lot higher than we originally had thought years ago. Then we also have, as I described, the Dichloromonas, which is can be a PAO and a GAO, depending on what the functions are. Now, based on my experience looking at DNA and microscopy, we have about I've done about 200 split samples comparing genotype to phenotype relationships, and kind of doing reports where we do micro and we do DNA, and we try to interpret the DNA as best as we can. And I would say it varies, but on average, if you're looking at community analysis, 16S rRNA sequencing, generally, if you have about 3 to 4% of a PAO per your total reads, that usually correlates with a pretty good population of PAOs and good performance. Now, the actual amounts are going to be very site specific based on how much phosphorus you have, what are the other DNA reads, things along those lines. So one of the areas of existing research is going to be, how can we grow certain types of PAOs, and what separates the growth conditions from, say, Dichloromonas, which is often very finicky, versus Tetraspira and Accumulobacter, which are much more consistent. So in troubleshooting a lot of the biological phosphorus systems, there are certainly times where the DNA can be an important part of it. Now, I do want to point out that there are other bacteria that are not classified as PAOs that do also have a higher affinity for phosphorus, a higher phosphorus storage capacity. So, for example, Microthrix is not a PAO by definition because it doesn't recycle phosphorus through anaerobic and aerobic zones, but it does store more by weight. So not every microbe is that 2% phosphorus, but technically for it to be a PAO, it has to meet the requirements that we're going to talk about. So just some basic terminology is aerobic would be a condition in which free and dissolved oxygen are available. Anoxic is going to be a condition where we have some form of combined oxygen. And anaerobic would be a condition in which there's no oxygen, whether it's free or combined. So in an anaerobic system, all of your nitrate and sulfate has been depleted. PHA is the intracellular energy storage of the polyphosphate accumulating organisms. So when VFAs or volatile fatty acids are taken up and stored, they're stored as PHA. So PHA is basically the storage mechanism for the bugs that we're trying to grow, the PAOs. So From a process control, we're taking up VFAs, and when they're store those, it's stored as PHA. So VFA, PHA, you can kind of think of those together. Some other terminology would be secondary release. This would be when we have phosphorus released when we don't want it to be. So anytime we have anaerobic conditions, we will release not only phosphorus, but also ammonia. But more important for the topic of this presentation is phosphorus. So there are times where you can get secondary release from anaerobic conditions, maybe in the final clarifier, or I would say more commonly, if you have too much endogeny. So basically, if the bacteria become starved out, when they die, their cell lysis and they re-release all the phosphorus that they've stored. So we would call this secondary release. The other thing in terms, and we'll cover the sludge age factor. But when you look at the different genera with PAO and the impact of sludge age, what we're going to find is a very high range in which enhanced biological phosphorus removal works. And it tends to be very plant specific. So I've seen plants that have successful enhanced biological phosphorus removal with sludge ages as low as 2 to 3 days where they're not nitrifying. And then also, extended aeration systems that can be as high as 30 or possibly even a little bit more day sludge ages. Advantages and disadvantages. When we have biological phosphorus removal, we are minimizing the need for chemicals, and that's an advantage. And it also helps to reduce alkalinity loss. So depending on where we are, I know in the northwest portion of the United States, there's a lot of areas where alkalinity is low. And when we nitrify and we convert ammonia to nitrate, we have significant alkalinity loss. A lot of the chemicals that we use to precipitate and remove phosphorus chemically also reduce alkalinity. So alkalinity is a buffering capacity of it measures the ability to resist a decrease of pH. So in general, once that alkalinity gets below 50 milligrams per liter, that's when we start to see a decrease in pH. And if the pH gets too much below 7, generally bad things begin to happen in terms of which bacteria are selected and treatment plan performance, things along those lines. One other quick note on alkalinity is when we look at nitrification, alkalinity is also what the nitrifiers are actually consuming. So their food is not the organic material, it's actually the alkalinity. So if alkalinity gets low, not only do you lose pH control, but you can also it'll cease nitrification at generally around that 50 milligrams per liter area. You also will reduce the extra sludge production from chemical addition. You're going to create iron hydroxide and other byproducts with like ferric or things like that. And then you're often going to get superior settling characteristics because the traditional PAO phenotypes are very dense and they grow within the flock and they tend to settle really well. So a lot of times the combination of having a designated selector zone and the PAOs makes for a better settling sludge. There are some exceptions to that, which we'll talk about a little bit later on within this presentation. Some of the disadvantages with enhanced biological phosphorus removal are the higher capital cost. So when you're retrofitting for BioP, you have baffles, mixers. You also have a lot of sensitivity to the any nitrate that may be coming back in your RAS stream. You have to look at the makeup of the actual treatment plant. What is your BOD to P ratio? And of that BOD, how much of it is readily available? And we'll talk about the different types of BOD and how it needs to be in the form of those VFAs in order for the bugs that we want in the selector zone to gain those competitive advantages and begin to store that food. So to summarize what's occurring is in enhanced biological phosphorus removal, we have a two-step process. So within our aeration basin, we have a generally, sometimes it's in one selector, sometimes there can be multiple selectors. And the idea is before your aeration basin, you have an unaerated area. And in that unaerated area, which we'll call the anaerobic selector, the PAOs, the bugs that we want to grow, they release orthophosphate as they're obtaining readily biodegradable organics or soluble BOD. So soluble BOD and volatile fatty acids are very similar. There's a lot of overlap. So a good way to look at volatile fatty acids would be if you think of the bacteria as having the cell wall, the cell membrane is kind of a filter, and that would act as their mouth in a sense. You look at the food that's coming in, and the food has to be small enough where it can go immediately into the cell wall. So basically the analogy would be smaller pieces passing through a filter. As you have septicity and you're creating volatile fatty acids would resemble those small pieces that are able to pass directly through, and they're very readily available so that the PAOs can take them up in the selector zone. So sometimes insufficient volatile fatty acids can be an inhibition. So you have to have the food, the BOD, but it also has to be in the right state. So there are some plants that have fermentation zones, areas where they're creating areas of septicity to help get that food in the right form. And then on the other end of the spectrum, you have some plants that have more BOD than they would care to have, and it can be a challenge. So to get BioP to work good, it generally there's a pretty broad range of different ORP selection or zones and targets that we'll talk about. So basically we have a two-step process. In the anaerobic selector, the PAOs are releasing orthophosphate, and they're taking up and they're storing food as VFAs. Now those VFAs, once they're stored, are then converted to PHA. So VFA, PHA, kind of think of those as the same thing in a sense. Then in the aerobic zone, the PAOs grow new biomass, and then they also take up phosphorus, and typically more than they release in the anaerobic zone. So a good example of looking at a profile for enhanced biological phosphorus removal would be, we would see perhaps like an influent of maybe 6 coming in a municipal system, and you're going to generally see about a 3 to 5 time increase of orthophosphate in that anaerobic selector zone if the conditions are right. So by definition, it has to release that orthophosphate in the anaerobic zone to be considered a PAO. Some of the criteria that you look for, and this is more on the engineering side, would be the BOD to P ratio, or the COD, which can be a little bit more complicated depending on if it's industry. And there's also a growing market for carbon supplementation. So different carbon sources, such as high strength waste, acetate, methanol, glucose, and molasses can all be used in terms of getting the conditions right, where your ORP values, which we'll talk about, are in the desired range for that activity that we want in the anaerobic selector, where we're storing food and we're releasing orthophosphate. So we described this, the role of volatile fatty acids. Think of it as readily available, kind of rocket fuel. And in terms of ORP, we have different ORP values in which we're going to generally aim for in different areas of the treatment plant. So the sweet spot for every plant tends to be there's not a hard number. So I've seen some plants where the ORP value in the selector can be minus 200 to minus 250, and that tends to work really well. And then there's others where sometimes it almost seems like you can't get it low enough, and it really likes it. And I've seen instances where minus 350, minus 375 is kind of that sweet spot. I think some of that comes back to the genetic diversity, and there's some of that we just don't really know in terms of which genera prefer what ORP values. There's also different methodology for testing ORP, so there is a little bit of times where comparing ORP depending on the actual measurement device can be a little bit not apples to apples, so to say. But in general, what we want in the anaerobic zone is that ORP needs to be low enough that we've depleted all of that combined oxygen, which can interfere with our reaction that we want. So the way that I like to look at it is negative ORP represents negative oxygen in a sense. So as you get lower on your ORP scale, you become more septic. And if you know the air and the risk of getting super low on that scale is the potential for food to bleed through into the aerobic basin, which can cause a whole series of problems, which we'll get into as we get through this. Sources of organic acids. So the food that we need for volatile fatty acids are those, the different sources that they can occur is at the head of the plant, lift stations. Anytime where you have wastewater that's not moving and there's no oxygen, you're going to begin these anaerobic fermentation reactions where you're taking those bigger pieces and you're fermenting them to smaller pieces. So a lot of times in industrial wastewater, they're just naturally present. So sometimes they're formed through septicity, and sometimes it's just a byproduct of the actual industrial treatment process. So equalization basins, primary clarifiers, all different areas where you can have septicity. A lot of times in the primary, if you are co-thickening your secondary sludge with your primary sludge, that's going to create, you're sending hungry bacteria to an area without oxygen, and that's going to create volatile acids. I apologize for that. Then also some of your sludge processing side stream fermentation tanks.
Sorry, I'm just going to interrupt here. I would just giving you a 5-minute warning. Sorry about that.
All right, sounds good. Hold on a second. I apologize for that. I have 3 rescue dogs and they're pretty crazy.
Sorry, Ryan, I'm just going to interrupt you again. You can actually just keep going. I just heard as long as you need. So keep it going.
All right, sounds good. So optimizing selector performance, we talked about every plant's going to be a little bit different in terms of where that is. So ballpark in the anaerobic zone, generally about minus 250 millivolts is where most plants tend to be successful. And then if you do have an anoxic zone, you need to have that at generally about minus 75. At that point, your nitrate becomes depleted. And once your nitrate and your sulfate are both depleted, then that ORP starts to drop lower. And then you start to get into those areas that we want, which would be kind of that 250 ballpark on average. So how do we control the ORP? We've got different tools, and these tools include our RAS rate. Some plants have the internal recirculation flow. We can control our DO sometimes, and then we also have the microscope. So in order to, if we want to get our ORP value higher, let's say our ORP is minus 400 and we've got a lot of BOD that's bleeding through, and in some instances to really get good biological phosphorus removal, in some instances, I found that they have to be pretty well stabilized sludge. And then in others, it seems like as soon as you get BOD removal, you're getting BioP. That comes back to what I suspect is probably the genetic diversity and some factors that are unknown. So there are times where it's got to be pretty well oxidized, pretty well stabilized where we see it. And then there's other times where, it's basically trial and error on the plant as far as what's going to be the most successful. So if we want to get if we don't have enough food and we have too much nitrate coming back, we can try to decrease our RAS rate. And by doing that, we're going to send less nitrate back if we're in a plant that's nitrifying, and then that's going to create less interference. So we have less nitrate to burn through before we get the ORP values to what we want. If on the other hand, the internal recirculation rates, if our selector is overloaded, we can send more and more nitrate. So we're using that nitrate and basically using the electron acceptor from the oxygen on the nitrate to get treatment. And ultimately, our goal is to get about 60 to 70% BOD reduction through that selector zone. So we don't want to have too much bleeding through. So we can control to a certain extent with our RAS. We can dilute it more, or we can make it stronger or more septic by having less dilution water through the RAS or less nitrate, which would interfere as an oxygen source. Also, if you have the DO too high, you can have DO carry you through the RAS, and if basically you're going to go through a series of utilizing dissolved oxygen, nitrate, and then sulfate, and then once those are depleted, then you're on to your storage. So basic schematic: you have your influent, your anaerobic selector, aeration tank, and then your return activated sludge coming back. So here's where we're storing food as the VFAs and releasing the orthophosphate. And now those bacteria have a lunch bag and they have a head start. And then here in the aeration basin, they're replenishing that phosphorus and they're taking up more than they initially released. And they have that competitive advantage because they already have their food present. So that's basically the cycle of what we're looking for. So this would be just a kind of example. This is a big extended aeration system. You can see they have an inner anoxic zone followed by aerated portions. You have other different configurations. You can have, it can get really fancy in terms of different places that you step feed. Basically, as long as you have those basic fundamentals of ORP, hitting that ORP values in the desired anaerobic system, anaerobic zone, that's really what we're after. So the other area where in some instances, if we don't have enough food, we talked about the carbon supplementation. So there are some plants that will look at in terms of if they don't have enough food, they can add more food through either supplemental carbon or sometimes high strength industrial waste, and that'll make it more septic and you can get that ORP to the zone that you'd like. There are some plants that have ORP control where they're interfacing that with a 420 signal and dosing that substrate accordingly to hit those target values. Every situation is really going to be very plant specific. So, biological phosphorus removal can be very finicky. And you can also make a case in some of the smaller municipal plants where sometimes the case for chemical phosphorus removal is there, that if you're using a low amount of chemical and you're able to meet the demands, sometimes that's an easier route. So it's going to be very case specific, and the engineering firms do a really good job of in general of vetting those different plants and kind of seeing what works. I have a picture here of a severe I&I incident. to symbolize our hydraulic retention time. But basically, when we look at ORP, the textbook's going to say 1 to 3 hours. In my experience, the hydraulic retention time is definitely a factor, but I would say ORP, getting that desired ORP value in the selector where you get that release is usually the number one priority in trying to optimize BioP. Now, when we have inflow and infiltration, that can really cause issues because that dilutes the wastewater. We don't have that proper concentration of volatile fatty acids. And generally what will happen is, unless you wash out solids, they just become inhibited. So you can have the PAOs there, but if they don't have the right conditions to do their cycling of phosphorus, then you're just not going to see the subsequent phosphorus removal through that. So it can be somewhat of a double-edged sword in terms of we need septicity, we need organic acids to obtain our biological phosphorus removal. But on the other hand, if we have too much and we have bleed through of the selector into the aeration zone, that can cause some undesirable traits like filamentous bulking, high dispersed growth, Zuglia bacteria, things along those lines. On the other end, if the selector is underloaded, that can be just as bad, because you can enter what we call no man's land, where you don't have enough. The ORP rate's not in an area where you are storing and you've depleted all your combined oxygen. So at that point, there's no treatment happening, but you're just creating further organic acids. So underloaded selectors can be just as problematic as overloaded selectors. So some plants have more flexibility. So depending on what you have in terms of your RAS rates, we've bypassed different selectors at times that were overloaded and step fed them and looked at different things to really try to get them dialed in as best as possible. So it definitely complicates what we're looking to do. So we talked about what's going on in the aerobic zone. The term for that would be luxury uptake of phosphorus. So for the PAOs to utilize the stored PHA for uptake of phosphorus, they need to be able to outcompete other bacteria for food in the aeration basin. So basically, they've already got their food, they enter the aeration basin, and then they're rapidly replenishing the phosphorus that they released. And we touched on secondary release of phosphorus. So any area that's septic is an area where you could potentially release phosphorus. So different things to be aware of when troubleshooting. Also, when we look at this diagram, a lot of our sludge handling side stream processes are, for example, like from an anaerobic selector, are going to be very high in phosphorus. So sometimes that will really distort our BOD to phosphorus ratio. So there are some times where you can have biological phosphorus removal, perhaps treatment of the side stream chemically, different things along those lines. So sludge aids in the impact, we talked about that. The textbook recommends 5. I've seen definitely less. And I would say an average of 5 to 30 depends on, it tends to be the sweet spot, and it's really plant specific. Now, one thing that a lot of plants that convert to enhanced biological phosphorus removal encounter is Microthrix and Nocardia or Actinomyces myclotofilament types. They both also have the ability to store food or VFAs in those anaerobic zones. So they're not technically PAOs because they don't release the phosphorus, but they do gain a competitive advantage. And the other thing is, when you look at the oil and grease concentrations, it's more so when that oil and grease is converted to long chain fatty acids like oleic acid, that those filaments have a better time outcompeting other bacteria for the fats, oils, and grease and that type of substrate. So when you introduce septicity to fats, oils, and grease, you create an advantage where some of those different filament types often begin to proliferate. So it's fairly common to have a plant, the oil and grease doesn't change, and you introduce a selector, and now you've created that septic environment, and now you have an area where you can have foaming or bulking from those various filaments. So that's gets into different plant specific troubleshooting methods that you look at. The other thing to be aware of, and this is why tertiary filtration is gaining popularity in a lot of places that have very stringent phosphorus limits, is when you look at the percentage of phosphorus per dry weight, the amount of TSS that you can get away with in the effluent. Let's say you have a phosphorus limit of 1. If you get over 15 milligrams per liter TSS, just with that and the stored phosphorus alone, you've probably gone over your permit. Land application considerations, you are storing more bacteria, more phosphorus in your PAO bacteria. So sometimes that's a factor that you may not have as much room if you're using land application. So I know we're just a little bit over on time. And again, I do want to apologize for being for the technical difficulties and signing in. I do want to share, this would be a reference book that I wrote. Also on TPO magazine website, there's a couple articles. I wrote an article on the interrelationship between PAOs and GAOs, and how that why that's important, and what that relationship looks like in terms of enhanced biological phosphorus removal. So with that said, I want to turn it back over, and I appreciate everyone's time.
Thank you very much, Ryan. Do we have any questions in the room? What are GAOs?
Oh, GAOs would be glycogen accumulating organisms. So they have the ability to store glycogen or sugar under those anaerobic conditions. So there is some thought that they can compete with PAOs, but they can also be complementary in some instances, where you can actually see. It's interesting that the traditional theory was always that they were in direct competition, but there's a lot of plants that have good phosphorus removal, and they have high populations of both GAO and PAO. So there's kind of an emerging thing there as far as that. So that's one of the areas of studies where we look at something like the dichloromonas, where it can be a variable GAO and PAO. And what we're really interested in is what causes it to behave one way or another. I suspect it's probably the ORP value in the selector is the most likely. But there's a lot of things that are kind of ongoing science, and we're learning about a lot of this as we're going as well.
Any more questions in the room?
Thanks. Just wondering if having the anaerobic and the anoxic retention tanks, does that create any additional issues with odor?
Generally, no. Usually for the odor to really be a problem, it's you usually have that air that's kind of introducing that into the atmosphere. So I mean, if it gets ripe enough, it can be, but it's not something that's super common. With that said, I think it always depends on who the surrounding community is. In terms of complaints and odor, there's, I can recall a lot of situations where we didn't really feel there was odor and we were getting complaints. So the introduction of the selector zones themselves, I don't think necessarily in most instances are going to be responsible for that. But you do have various odors that are associated with different organic acids, which you're forming in those zones. So the answer would be kind of, it depends. In some instances it could, but most of the time, usually not, would be my answer.
We have one question online, I think.
All right.
Can you just switch the screen so we can see that question, or will it turn off, Ryan?
I think you have the screen.
Oh, Ryan, can you provide a copy of the slides? Are you able to do
Oh,
that? after?
yeah, I think I sent a presentation over, but I will after this I'll send something so everybody has the presentation.
Awesome, thank you. Is there any more questions in the room?
Just wondering where you're, and after me, there'll be one more question. Wondering where's your book available?
Oh, sure. If you go to my website, the book is available on Amazon. My website is Ryan Hennessy Wastewater Microbiology. I am in the process, and I'm hoping within the next hopefully couple weeks it'll be available on open access through IWA. Right now it's available on Amazon, so there's a link on the website there. If you just Google or the internet search of my company, then you'll access the page. So that book is actually basically intended to be a follow-up for the 3rd edition manual that Dr. Jenkins and Dr. Richard wrote in 2004. Dr. Richard was my mentor, and I was very fortunate to learn under him for 10 years, and they never got around to making a new edition. So the book that I wrote is taking the information from that 2004 manual, incorporating the DNA information and some other things that have changed, and just giving it basically as good of a relevant update as we can.
I just had a question about the ORP.
Mhm.
So when you're in the 400s, the negative 400s with your ORP, and you're running out of a fermenter to do dewatering, how is that affecting the phosphorus removal with your PAOs?
Okay, so if basically your effluent phosphorus is going to really tell the story as far as success. So your target ORP value is going to, it might be as low as minus 375, or it might be minus 200, minus 225, depending on where it likes to be. So when you have a fermenter, generally, if you have the longer you store that sludge in the fermenter, the more ripe it gets and the more VFAs you create, and therefore the more septic and lowering the ORP value. So if you have a fermenter in place and your goal is to increase the ORP because it's too low, then you'd want to have that sludge pumped more frequently, and then vice versa. If you don't have enough food and you need to ripen it up, then you would try to leave that sludge in there longer to create more VFAs. So your ORP is, you could somewhat correlate your blanket depth of your fermenter to your ORPs, and then also taking into consideration your performance and kind of where you want to be.
Anyone else? All right, thank you very much, Ryan. That was great.
All right.
A round of
Appreciate it.
applause.
All right, everyone I hope everyone enjoys the rest of the seminar and have a good time.
Thanks, Ryan.
All right, thank you very much.
When do you want to? We'll start the Jeopardy at